The one thing I did want to mention is Corey Angeroth is moving on to greener pastures, for the lack of a better analogy. And so Ryan has agreed to take his place on the tag. So good luck to Corey, and thanks, Ryan, for volunteering your services to help us.
Hey, John.
Yeah.
Hey, so this is Corey. So yeah, I just hopped on here for the beginning of the meeting to say thanks to everyone for all the years of working together here with you on Great Salt Lake. It's been a privilege and an honor to work with you all. And Great Salt Lake, it's a great place to work, and I really enjoyed my time working here. So, as John mentioned, I've accepted a position with the USGS headquarters, basically. Fortunately, I get to stay here in Salt Lake City. Don't have to move anymore. I guess that's one of the positives of the pandemic, maybe. I don't know. That people can accept positions and not have to move to DC. So so I'm actually going to be working on the I guess my title is the Deputy Coordinator for the Groundwater and Stream Flow Information Program. So on our stream gauging, our national stream gauging program, and working with Congress and other stakeholders on that. So there you go.
Well, thank you. I appreciate you helping us load these many years after Dave left. So thank you a lot, and we appreciate all your help and look forward to having Ryan pick up where you left off. So,
all right. Thanks, John. Thanks, everyone.
Yeah, take care, Corey. So in addition to, and just because we were making that change, I think we've had John Cavett has been our avian representative on the tag team in the past. And as you probably have all noticed, he hasn't been at any of the tag meetings for quite some time. And so just to do a little house cleaning, we decided to upgrade and or provide our newest tag member, Chris Klein, for the avian representation. So she's going to be taking over for John Cavett, and Ryan's going to be taking over for Corey. So that way we maintain our group and also the representation on the group. So just wanted to update everybody on that. And then I hope that this will be our last meeting via Google or whatever, and we'll be able to meet in the future, providing everybody is comfortable with that. We'll be meeting in person, but we will try to still provide a online option. That way we can ensure good attendance, and that way if somebody or anybody is a little apprehensive about coming, we can still have you here so you can participate. So with that being said, if nobody has any questions, I'm going to go ahead and transition and let Ryan start with his presentation on the research update. Very good. Ryan?
The good news is Corey is going to keep working in this office, so it's not like we're in this location. So we're not going to lose his historical knowledge. So, but anyway, we look forward to moving forward with our Great Salt Lake team, and that includes myself, Christine Rumsey, Scott Heenick, who I don't think many of you have met yet, but he's a chemist. And there'll be others, David O'Leary, for example, who's our investigations chief. So we've worked hard to kind of build our Great Salt Lake team up, and so we're feeling okay moving forward. But saddened that Corey is taking his new role. So Christine and I, Christine, do you mind bringing that presentation up, sharing it? We'll get started.
Can you see that?
Yep.
Great.
Looks good. Okay. So today you'll hear from myself, Christine, and Molly Blakowski. We're going to tag team this. Christine's going to run it. So we'll give you an update on our work out there. Christine, next slide, please. Okay, we're going to talk about lake elevations. Corey likes to always start these tag meetings off summarizing where we're at with our elevations. Then we'll move into stuff that is funded by the Great Salt Lake Ecosystems Program by John Luff. So we've got inflow monitoring at Goggin Drain and Bear River Bay, inflow and open water nutrient sampling and analysis, and then nutrient mass balance and cycling project update. This is the big effort that Christine and others have headed up. This is a project to get more at the flux of nutrients between different nutrient pools. So we'll get a good update on that. Next slide, please. All right, let's start off with the elevation of Great Salt Lake measured at our gauge at the Great Salt Lake Marina. We call it the Saltaire Boat Harbor. So, you know, right now we're at 4192.8. That's up about 0.6 feet from the last tag meeting in early December. Let's go ahead and hit next, Christine. We're about 1.7 feet down from a year ago. Next again. One thing that's kind of concerning about this year relative to previous years, you know, usually in the winter and spring, we like to see a good uptick in lake elevation. So on this plot, we're showing about a 1.2 foot increase for this time period a year ago. This year, we're looking at about 0.6 foot increase in lake elevation. So we're about half of where we should be. If we think around a foot and a half is a reasonable increase in the fall and winter winter and spring. So again, this is just another little warning to everybody that we're obviously in quite a drought cycle here, and we're seeing the consequences here at Great Salt Lake. Next slide. And then this is just the whole period of record going all the way back to the 1800s. And, you know, it's the take-home message for the group is that we've been in a low water paradigm for the last 15 plus years. And we're just kind of fingers crossed right now. We know that we've got a very dry, a relatively low snowpack right now. All the predictions for runoff are that's going to be well below normal. So this is something we're going to all have to keep our eyes on moving forward. Next slide, please. All right, here's the lake elevation data for the north arm of Great Salt Lake. This is our gauge near Saline, Utah. We're at 4192.3 feet today. That's up about a half a foot from our last tag meeting, and we're down about a foot and a half, similar to the south arm from a year ago. And we're at about a 0.5 foot head difference between the north and the south arm. And we were about 0.4 feet head difference for our last meeting. So that's kind of go ahead and hit next, Christina. We can cycle through some of these images. So this shows basically a year's worth of record at the site, and it demonstrates that 1.5 foot drop. And this shows a longer period of record here. Again, I want to just emphasize that winter spring recovery that we're not really seeing this year, or at least we're not seeing to the magnitude that we've seen in previous years. So in January 2020, if you're looking at that plot, you see the relatively large increase in lake level. And then if you look at January 2021 to see where we're at right now, we're just going to keep a close watch on that to see if we can bring the lake level up. But like I say, the forecasts are not that great right now. Next slide, please. That's our period of record for the saline gauge. Our current water level, if you take out the closed off culverts, which didn't allow water to move from the south to the north, our current lake level is pretty low, maybe the lowest in the period of record. You see some slight, lower elevations back around 2016, 2017. That's related to those culverts being shut off. So we got low lake levels out on Great Salt Lake. Everybody knows this. Next, please. Okay, so we're going to talk, we'll just do a quick reminder to everybody where our monitoring sites are. So we have continuous flow gauges at the Weber River, and the locations are shown on the map. Goggin Drain, which John left of the Ecosystem Group funds. Great Salt Lake Breach near Lakeside, which is the old breach. And then we do periodic flow measurements at Bear River Bay outflow and at the new breach on Great Salt Lake. And we all know that flow data is the foundation for water and salt balance and nutrient balance monitoring of the site. So just kind of take a quick look at that, the site location maps, the map there, and make a mental image of where these gauges are. So when we look at some of the data in the next slides, you'll have an idea of where they're actually located. Here I'm showing, go ahead, yep, next slide. So here I'm showing the annual flow volume in acre feet that has passed by our gauge on Goggin Drain for the last 15 or so years. So the blue dots are the amount of water in acre feet for a given year. The dashed line is the average, the long-term average for this site, which includes data from 1964 to 2020. So just glancing at this plot, most of the blue dots are below that average line for the last 15 or so years. We have not had very good, water years. We've had a few good ones in the mix, but this builds into the story of why we're where we're at with the lake elevation data. Next slide, please. And here I'm just showing you that we are completing those periodic discharge measurements at Bear River Bay outflow. We do not have a continuous monitoring stream gauge there, but we perform these periodic measurements to correlate and compare to the upstream continuous gauge, which is Bear River Corinne, and we're going to look at that in just a second. But each time we go out and make a discharge measurement at this site, we also collect nutrient samples. And so that all gets built into the nutrient mass balance work that we're doing on Great Salt Lake. So I'm just showing with this slide that we're completing those periodic measurements at the Bear River Bay outflow. There's not as many measurements as you might think there should be, but that's often because there's no flow at that site in the summer during the irrigation season. So just something to keep in mind. Next slide, please. So if we want to kind of look at a continuous flow record for Bear River Bay, the closest gauge to Great Salt Lake is the Bear River near Corinne, Utah. You see the location in the upper right on the map. And I'm showing here the annual flow volume in acre feet. This is the exact same plot as we showed for Goggin Drain. The long-term average flow volume for this site is developed from a record that goes from 1950 to 2019. That long-term average is about 1.17 million acre feet. And just like Goggin Drain, you can see most water years, the amount of water passing that gauge are well below that long-term average. We had a good runoff volume in 2017 and 2011, but other than that, we're basically barely at average or well below average. So hence the low lake levels in the lake. Next slide, please. Same plot. This is for the Weber River near Plain City, Utah. I forgot to put a little reminder location map in the corner of the slide, but most of you know where it's at. So again, there's the long-term average data shown by the gray dashed line that's got record from 1949 to 2020. That long-term average value is about 322,000 acre feet. For the last 15 years or so, we've been, most of the time, we're well below that on a year-to-year basis. We've sprinkled, there's been a couple of 2-3 good years in there with above average flow. So there you have it. That's kind of the take-home message for why we're seeing such low lake levels. Next slide, please. Okay, so we do these periodic measurements at the new breach, which in my mind is one of the top 5 most important monitoring locations for our office in the state of Utah, because we know how important that new breach is to managing the salt balance in Great Salt Lake. So we make these monthly flow measurements during calm conditions. We've done that since December 2016. And one thing we're doing to kind of push the envelope at this site is to get a resistivity sensor string that we can put in the water column to delineate more precisely where the interface is between the north to south flowing water and the south to north flowing water. So basically the boundary between the north arm water and the south arm water. It is a bidirectional flow site. It's very important that we're able to pinpoint that location in the water column because then we'll be able to more accurately measure real-time discharge at the site. So we've been working with the hydrogeophysics branch of the USGS. They've been doing this pro bono. They've built a $10,000 sensor string. That sounds, the sensors are spaced basically 10 centimeters apart. So we're going to have just really good resolution. And we're planning now to get that thing deployed here over the next month or two. We're working, in coordination with Utah State University at this site. They're working on a numerical model. So we're engaged in conference calls and site visits with that research group. So there's real exciting developments in the very near future for this site in terms of being able to accurately quantify water moving back and forth. Next slide, please. All right, so this is just the monthly manual discharge measurements that we make at this site. So we have discharge in cubic feet per second on the y-axis on the left side, and on the right side of the plot, I'm showing the elevation head difference between the north and the south arms of the lake. The blue circles are south to north flows. The red circles are north to south flows. Just looking at the most recent data, the amount of water moving from south to north is roughly 1,000 CFS. The amount of water moving in the opposite direction from north to south is around 200 or 300 CFS. These values are not outside the range that we've seen historically. And y'all know kind of the story when you move to the far left of the plot, why the blue values plot so high. That's when the new breach was opened up, and we had a lot more elevation head. The south arm was quite a bit higher than the north arm, so there was a lot of water that moved through there initially. So the interesting things in this plot is you look at the light gray line, which is the head difference. You see how the light gray line and the blue circles sort of match up based on simple head difference between the two halves of the lake. And so this data also very critical to USU in helping them develop their numerical model. Next slide, please. Okay, so we are sampling the freshwater inflow sites for nutrients and also the new breach and the breach at Lakeside. So that's 6 sites. We're sampling those about every 6 weeks. That effort began in 2011, and this is all part of an effort to quantify the nutrient mass in the water of Great Salt Lake. And so if we can move to the next slide. And Dave Naffs has, he was our research geochemist who worked on this, who kind of developed this monitoring program. He published a paper on the nutrient mass balance in Great Salt Lake in 2014, and he's now working on extending that adding record from 2015 to 2018. I showed an example. Actually, that slide is next, but he's currently still working on this, even though he's retired. He's done the nitrogen inflow loads for those inflow sites. He's working on the monthly nitrogen pools in the north and the south basins of the South Arm, and he's also going to compare the nitrogen pools to his previous paper, and then also he's going to go into detail on the impact of the new breach opening on nitrogen distribution in the South Arm of Great Salt Lake. So that's what he's working on. I think the next slide is one that we saw at the last TAG meeting, where he's computed the nitrogen loads associated with inflows, the freshwater inflows, and we discussed last meeting how the Bear River inflow is the largest source of nitrogen to Great Salt Lake. So he's continuing to work on this for us. Next slide. And then we have open water nutrients sampling, which goes into the work that Dave's doing. That the bulk of that sampling is taken care of by John Luff's group, which we very much appreciate. That's a lot of work that they undertake to get these samples. They sample 4 sites at 2 different depths, so a shallow and a deep depth. They're doing that every month. So in addition to the nutrient analyses that we get, we also measure density, and via the equation of state, we can get at salinity. Next slide. And just quick example here. So here's the density values measured at 2.5, GSL 2565 and GSL 3510. 2565 is a little closer to the breach. It's in the northwest portion of the south arm, and GSL 3510 is basically in the center of the south arm. You know, we've seen this plot many times, so I'm not going to explain. You know, go into detail some of the previous data. We all know that you see a divergence in circles and triangles due to the existence of the deep brine layer. So just looking at the most recent data at the far right side, the circles are for deep water sites, or excuse me, deep water samples, representative of the deep brine layer. The triangles are shallow water samples. And looking at these values compared to historic values, we're within the range we've seen over the recent past. And there's some seasonal, you know, increases due to declining lake levels. I would just point out that the density values that we're seeing for 2565 and some of the more recent density values for 3510 are on the high end relative to the historic data that we have, or at least the recent historic data going back to about 2011. So it's something we'll keep our eye on. Next slide. I'm not going to go into great detail on these. This is just to demonstrate that the nutrient sampling is happening. This is data for GSL 2565, and we're only showing total phosphorus and total nitrogen. We have analyses for other nutrient species. The shaded areas on here represent when there is a deep brine layer and when there's not. The circles are deep water samples, or the dark blue circles are deep samples. Shallow circles, or excuse me, the turquoise are shallow samples. And we see that divergence where we have higher nutrients in the deep samples relative to the deep brine, or excuse me, shallow water. And this is this is information that Christine is really wrapping her mind around to help with the nutrient flux work, actually. And then, of course, Dave Naff's is using it for the nutrient mass balance work that he is doing. Next slide. This is the same data, total phosphorus, total nitrogen for GSL 3510, and same story. The deeper water having higher total phosphorus and total nitrogen values relative to the shallow water. Next slide, please. Okay, I blew through that pretty quick, but we have a ways to go yet. So I'm going to turn it over to Christine.
Hey, real quick, Ryan,
Yes,
before Ann had a couple of good questions. I was wondering if you could address those. One, I am curious about, and mostly a matter of curiosity about whether or not the high flows have shifted, and I would expect that through observation they have shifted to an earlier time. And then she also mentioned that there's going to be a new discharge. I actually can't remember what year that will occur, but she's right in that will be directly going into Gilbert Bay rather than Farmington Bay. So if you are sampling at the causeway, then you are going to miss that, or there'll need to be some way to address that flow out there.
Yeah, it's a fantastic point. So yeah, we're gonna have to wrap our minds around how to deal with that, and you know, hopefully with the open water sampling, you know, we'll be able to track just the overall impact of that. But yeah, that's a great point.
Have you seen a shift in high flows at all, timing wise?
I personally have not taken a close look at that for Great Salt Lake. So that's a great question. I think everybody knows that overall, you know, I don't even know. Can I say climate change? Let's just with the climate, the interesting climate that we're in. By the way, the federal government's not telling me not to say climate change. I just want to be sensitive to everybody who's on this call. But I think we all agree that there's interesting things going on with climate, and I think one of the things people talk about being a consequence of that is the runoff happening earlier each spring, and we haven't quantified that yet, but my anecdotally, I can tell you the last few years, yes, it feels that way, but we have not done a detailed analysis of that in the Utah Water Science Center. Other USGS offices may have.
Yeah, I would guess the same thing. Also, GSLEP purchased some equipment to verify the status of the lake. It was, maybe you've heard of it, it's called a Magic 8 Ball, and we asked it what was going to happen to the lake, and it said outlook not so good. So it verifies your data, I believe. So
yeah, I mean it's pretty startling to look at the inflows for like Bear River and Weaver River over the last 15 or so years. That, yeah, we all hope for a good winter this year, and it just doesn't look, it's not playing out. So
yeah. Hey Ryan, this is Chris Klein. Just quickly, is there kind of a reason why the nutrients in the deep brine layer are, so much higher than they are in the shallow water? And then I guess Ann wants to know why they fluctuate more than they do in the shallow water, and they fluctuate more in the deep brine layer.
I mean, the nutrient values are higher in the deep brine layer because I think as material settles out and it hits that higher density water, it kind of gets stuck and it slows down. You know, it takes it a while to settle and make its way through the deep brine layer and then into sediment. So I think it just kind of, the nutrients accumulate kind of at that density boundary, and then it just has a longer residence time in the deep brine layer. But that's just my opinion. This is all kind of why Christine and others that you see listed on the slide that's up are doing this kind of more detailed investigation to understand exactly what is going on with the nutrients. But I suspect it has something to do with matter getting sort of stuck in that deep brine layer, not settling out.
Would it be a matter of algal uptake? So basically, you've got algae in the upper water column that's actually taking those nutrients up to grow.
Absolutely, yeah, okay. Sure, yep. And again, that gets at why, that's exactly why we're doing this more in-depth nutrient mass and cycling study.
Great. Okay, thank you.
And maybe looks like Krishna had a comment about doing a month. So maybe next tag, if you could provide a monthly time series analysis of the flow, that might be helpful.
Oh yeah, yeah, sure, absolutely. Yeah, we can do that. And Krishna, please feel free to reach out to us anytime here too, if you want us to provide you some up-to-date information, and I can also show you how to access that data online. It is available online to everybody. You can get monthly volumes of water, passing gauges, and acre feet for all of our continuous flow sites. So.
All right, great. Well, Christine, it looks like you're up.
Okay, hi everybody. I'm just gonna give a quick update of our nutrient project that we launched this summer. Myself, along with help from Molly Blakowski, who will be talking about kind of a subset of the project. So just to remind folks who maybe have not seen our work before, I'll just say briefly, our objectives for this study are to collect and quantify nutrients from a variety of pools in the Great Salt Lake, kind of as you can see here, from sediment, ceston, brine shrimp, the water column, etc. And then the whole idea, the big goal behind this is to just to better understand the processes controlling nutrient availability in the system. And so in the December meeting, I kind of went through all, our sampling effort, what we had done in August, walking you through kind of the sample collection and maybe some about the sample processing, how we generated a lot of new samples. We're going to be generating a lot of new analyses to kind of pour through. And so again, that's done, and we're still sort of working on just wading through the data. We've been getting a lot of data back from labs. We're still tying up some sample processing and sending samples off for analysis. Had some struggle with labs in COVID. So kind of wrapping up the lab and the analytical work, and now really beginning to pour over the data, organizing it into a usable form, and then checking for quality assurance to make sure the data are robust and usable. And so that's our focus at the moment, is really putting all that into what we call a data release that will be available publicly and published online, so all can access it. And then that will complement our the analysis and interpretation that we're going to write up in the journal article. So just to give you an idea, we're very focused on data at the moment. And as part of that, we dug into a couple components of the data we have collected. So I'm going to just share a couple of things with all of you that you might find interesting. The first is that I'm going to share data from our upper brine layer, deep brine layer interface work, where we collected water across that mixing zone. And then Molly's going to talk about preliminary results from the atmospheric deposition of nitrogen. So, all right. First part, capturing the upper brine and deep brine layer interface. So this was work proposed by the TAG, essentially. This is something that you all were really interested in, was trying to refine how the chemistry of the water changes as we go from the upper brine to the deep brine layer. And so this was a challenge to try and figure out how to do. So we found this equipment that is typically used in groundwater, and we just strapped it to an anchor and a submersible buoy and deployed it in the lake. And as you can see, we knew right away, it was really exciting when we pulled up this anchor. We knew right away we had captured it. You can see we have this clear water in the upper brine. And then as you go down deeper into the deeper layers, you see that brown color that is typical of the deep brine layer. So this was successful, and I'm happy to share some preliminary data from these samples. This was done at 2 sites in Gilbert Bay. 3510, which Ryan mentioned, is in kind of the central part of the South Bay, and then GP14, what we call GP14, is in the southeast portion of the lake. So I kind of wanted to just start off by putting these sample locations in the context of the entire lake profile. So here I'm showing a set of plots. The top panel of plots represent data collected at 3510, and the bottom panel represent data collected at that southeast site at GP14. And I'm showing data for 4 field parameters: temperature, specific conductance, pH, and dissolved oxygen. And along this top x-axis are the parameter values, and then the y-axis is just the depth. So kind of 0, think of that as the water surface, and then we're just kind of going down through the water column, through the upper brine, mixing zone, and deep brine layer, and then kind of this bottom, that's the sediment, is this brown kind of box there. So the full profile of data, that was collected with our in situ sonde, just like every meter, and then when we got down into the mixing zone, we did like every 20 centimeter resolution. So we can see how, the gradient of change, in this case for temperature, and then the black dots represent where we collected our samples. So where these actual water samples were collected along this profile. And what's exciting is, we captured it. We captured the mixing zone. So we have a couple of samples, an upper brine, a couple throughout that mixing zone, and then in the deep brine layer. And then this orange line is kind of just showing the gradient. So the change in this parameter value between the sample bottles, or between the sampler locations. And so this really helps us begin to characterize what that interface looks like. In the case of temperature, we can see the majority of this change, and the gradient is on this lower x-axis. the majority of the change is happening over like a half a meter distance in the case of temperature at 3510. In the case of specific conductance and pH, there's a huge amount of change happening within about a 0.2 to 0.3 meter distance. So this is just fun to look at and just see, okay, what does this mixing zone actually look like, and over what distance is it actually occurring? Another interesting observation is that dissolved oxygen, it changes over about a half a meter distance, but it's actually shifted upward. So we see a drop in oxygen about a half a meter above what we might call the mixing zone as identified by specific conductance and pH. So this is kind of an interesting finding that might, begin to suggest the processes that are active, whether that's, bacterial respiration, or maybe it's kind of the end of the photic zone, so there's no photosynthesis taking place. We're not sure at this point, but an interesting observation. All right, and similar characteristics were observed at GB14, where we see, again, that gradient. We were able to capture that with our SNAP sampler assembly. So moving on to the nutrients. So now with each of the water in each of these samples, we analyzed for a variety of constituents: nitrogen, phosphorus, their species, carbon, sulfate, and salinity. And this was just to really begin again to try and better characterize how nutrients might be moving across this interface. If we look at the salinity plot, we can again see how we captured the zone of change. Again, and I sort of truncated the axis here. So we're kind of zooming in on this deeper water from 5 to 7.5 meters deep. So again, going from upper brine through the mixing zone into the deep brine layer. And so nitrogen is pretty fun to look at here. So I'm plotting different species, including total nitrogen, total dissolved nitrogen, organic nitrogen, ammonium, and nitrite. And so if we start looking in the upper brine, we can see that most of the nitrogen is dissolved and most of it is organic. As we move through this mixing zone, the interface between upper and deep brine layers, we can see how all the species increase. So we have higher concentrations of total nitrogen, of total dissolved nitrogen, organic nitrogen. We also see ammonia show up, and then those patterns continue into the deep brine layer. We also observe that there's a much greater fraction of total particulate nitrogen in the deep brine as we move deeper. So this is kind of fun to just begin to try and think about how nutrients are being, or nitrogen in this case, is being transformed, how it's being changed between these two different waters. Inorganic nitrogen in the form of nitrate and nitrite was not observed at any of these depths. So similar patterns for phosphorus, where we have here we're plotting total phosphorus, total dissolved phosphorus, and orthophosphate. Most of the phosphorus in the upper brine is dissolved. Most of it is organic. As we move through the mixing zone, we see a greater fraction of organic phosphorus and just increasing concentrations of basically everything. Carbon, same thing. We actually observe more dissolved organic carbon than total particulate carbon. Particulate carbon certainly increases as you get down into the deep brine layer, so that kind of matches what we see both for nitrogen and phosphorus. And so what we hope to do with this moving forward is to really get find a variety of ways to estimate the flux. I think that's the main question, is how, we have a high concentration of nutrients in the deep brine, how much is getting up into the upper brine, and what is the rate of that flux? That's going to be, rely on a variety of assumptions, and we're going to estimate a diffusive flux. But with these kind of higher resolution concentration values, we hope to get a better answer to that question. And again, these similar patterns were observed at GB14. So yeah, I will now pass it on to Molly. So she's going to talk about what she and Dan Jones of our office did to quantify the rate of atmospheric deposition to the lake. Molly and Dan really kind of took the lead on this subtask of the project, and we're super grateful to have both of them involved. Molly's actually a PhD student with Janice Brainey at Utah State, and she is studying the composition and transport of dust from Great Salt Lake Playa. So really happy to have Molly. Go ahead, Molly.
Hi, can you hear me okay?
Yep, can hear you.
Okay, that's great. I apologize for any connection issues. You guys have been going a little out and in for me over the past few minutes. So I'm going to just really briefly talk about what Dan and I have done to contribute to this nutrient cycling project. We deployed atmospheric deposition samplers at 9 sites around the perimeter of the lake because the goal was to try to capture a regional signal that could be interpolated across the lake. We selected high elevation sites that are far away from roads or other sources of potential contamination. The elevation of our sites and the height of our samplers reduce the chances that local material not representative of regional sources would wind up in our samplers. So whereas when I'm sampling on the playa, I'm looking to get material that's generated directly from the playa so I can understand the composition of those sediments. Here we're trying to capture a regional signal of dust and aerosols that can be dispersed throughout the lake. So Andy Carlson built some really excellent sturdy sampler frames with metal rings to deter birds from getting too curious about our interesting looking samplers. These are funnels that are sitting within the metal rings, which was a pretty genius suggestion from Janice. The rest of our sampler design was also modified from samplers used in Janice's previous work. So we have funnels filled with marbles, which increase surface area for particulate matter to stick to. The sample bottles are clamped to a piece of tubing that's separated from the funnel with a piece of mesh. This is to keep bugs from falling into the sample bottles. So we're going to give you just some really preliminary results for nitrogen that we're kind of excited about. Christine, you can move to the next slide. Unfortunately, we did lose some sample bottles in the crazy windstorms we had over the summer. So we had, the good fortune of conducting an atmospheric sampling campaign during a historic windstorm and crazy wildfire conditions. But we still were able to analyze samples from Lakeside, from the north and south end of Stansbury, Unicorn Point at the south end of Antelope Island, and a few other locations. And by using these few data points, we were able to come up with a rough estimate for atmospheric nutrient deposition across the lake. So here I'm showing nitrogen. We've interpolated the data that we were able to gather for these sites across the lake using inverse distance weighting interpolation in GIS. So you can see that we have higher values over from areas that are closer to higher land use. So Buffalo Point is our highest spot in terms of nitrogen. The values you see here are reported in annual fluxes of nitrogen derived from atmospheric deposition. So what does this mean and how does it compare to previous results? Christine, you can move forward to the last slide here. The previous estimate from the work by Dave Knapp's estimated that atmospheric deposition of nitrogen to just Gilbert Bay is about 564 metric tons for the year of 2010. Our first pass at adding up values from this interpolation across the lake is just over 1,500 metric tons, and that's to the entire lake for this 2020 snapshot. So we had our samplers deployed for about 8 weeks. We're continuing to refine this estimate in the next steps of our spatial analysis. So considering that there might be some extra local influence to sites that are closer to high land use areas. But we're pretty excited that we were able to, haul those huge samplers up to these high elevation sites. And even with a small amount of data, try to make a meaningful estimate for atmospheric deposition of macronutrients to the lake. So that's all I have.
Thanks, Christine, Molly, and Ryan. Anybody have any questions for the USGS team? All right then. Well, I will also mention that if you are sharing a computer, will you type in your name and affiliation just so that we get an accurate depiction of who's here, so we can put that on our notes. So if you can type that in or send an email to John Neal, that would be great. And then Kyle will go over the shrimp demographics. Hopefully. Go ahead, Kyle.
There we go. Okay. Now you should be able to hear me. All right, so this is the brine shrimp demographics from our last update from the tag in early December through the present. So we're really just going to be updating the last 4 or 5 sample runs in January and February. So all of our graphs are going to follow this similar outline here, where the table on the left side of your screen is the monthly averages, whereas the lines in the graphs are going to be the black line is the 20-year average. The red line is the current year, and we're showing both graphs for both 2020 and the new data for 2021. And then all of the rainbow colored lines in the background show the extremes of each year, so that way we can see where our data falls as far as the average and the highs and lows that we've observed over the last 20 years. So looking at water temperature, nothing too out of the ordinary moving into 2021. We're just below average, a little cold. Starting to warm up a little quicker here in February. You can see the average was about 3.1 degrees. And we expect to see hatching on our next run. We were not able to get out this week due to the weather that we're experiencing right now with some of the winds. So hopefully we'll have some new data next week, and I expect to see some of our nauplii starting to show up, but we're not really sure that that's going to happen. Looking at salinity for this current time, we do see that 2020, followed that traditional trend of lower salinities in the spring with that runoff time, moving into higher salinities throughout the year. Currently, we're starting above average in 2021, hoping that we'll get a little bit of runoff and bring that salinity down out of this higher 15, almost just over 15% for the monthly average there in January. That's starting to get in that realm of concerning, but it is starting to come down. Looking at the chlorophyll A values for the last few sample runs, in November and December, we start to see the brine shrimp die off, and we'll talk about that in a moment, but we start to get that real good rebound in the phytoplankton community, and we start to see our chlorophyll A values really start to rise through November and continue rising through December. And then we see this really sharp decline through January, but it has since rebounded in February. And this was something that was a bit concerning to us at first, just because there's no brine shrimp out there. There haven't been any brine shrimp out there. Where did the chlorophyll go? And we looked at some of the previous years, and we've seen this pattern occur before, and also looked at some of the cell counts that Gary performs for us, and the overall proportions of phytoplankton types did not shift, but there was a decline in the overall cell counts. So there was a bit of a phytoplankton die-off for some reason. Could be attributed to a combination of factors, the cold weather, the higher salinities. We're not really sure, but it's not too concerning because we do see this quick rebound. And as we'll talk about here in the next couple of slides, this is looking back at the same information for chlorophyll A for 2011 and 2012. And you can see this really sharp decline in February of 2012 on the graph on the right, to where we dropped from 17 micrograms per liter down to about 5 micrograms per liter. And then we see just a real sharp rebound in March. So this is something that has occurred before. It's not too concerning, but it was just something a little different. This is another look where we saw this same pattern in 2014 and 2015. And you can see kind of that similar dip there in January of 2015. Again, the graph on the right, where our January numbers just take a small dive, even though there's no brine shrimp present. So again, quick rebound, and then starts the year off moving forward. So looking at the cyst densities, 2020 remained above average pretty much all year long. 2021, again, we're starting off a little bit higher than average, a little bit higher than our preferred 21 per liter, sitting at about 40-ish cysts per liter right now. But again, nothing terribly concerning. It's just likely we'll be on the higher end of things going into the spring. So looking at the peak cyst numbers, our peak did occur in October. You can see that at 187 per liter. If you remember from the August tag last year, we did, our prediction showed with the removal of any of the higher sites, our prediction was 174 through 115 with an average of 145. So this peak at 187 was a little higher than we expected, but that just shows that we had either a difference in survivorship early on, which we did see early on with the juveniles, or we had some other factor where it just wasn't as much of an impact as we had originally predicted. And so we ended up with a little bit better of a cyst production than we would have expected, which is a good thing. So hopefully we'll see something similar this year, and we should be just fine moving into the spring. We don't have the data to make that prediction yet. We'll have that coming up in August, just because I want to make sure that we capture that original hatch of the knobly eye. We haven't seen that quite yet. So the rest of these graphs are going to be quite boring, just as an update from December, because everything started to die off in November. You can see that the values on the right, our monthly average for most life stages was 0 in November and remained 0 through February. So we just haven't caught any hatching yet. So this holds true for all the life stages. For juveniles, everything was gone by November and nothing has hatched yet. Similar with all the different stages of adults. And I think if anybody has any questions, this is pretty much going to be the bulk of my topic. So go ahead and open those up.
Does anybody have any questions for Kyle on the demographics?
Just a point, this is Gary Belovsky. It's just a point, Kyle, I took a quick look at years that we saw a decline in, January, February in the chlorophyll counts. And I tried to see if there was some relationship with temperature, and it didn't seem like I could see an obvious temperature link there.
And we did something similar, and I don't know that it's just based on temperature. It's got to be a combination of multiple factors that we're just not able to pick up.
Yeah, I just thought, that would be a big signal that we, a strong signal if it was there and I didn't see it. So,
yeah, that looked like the strongest signal to us too, and we couldn't find anything either.
All right. Well, if nobody has any questions about the shrimp, then we'll move on to Gary's presentation. And hopefully you can share your screen and don't have a issue.
Can everybody see that? No?
Not yet.
Okay. Now?
Nope.
Okay. Let's see. Got it now?
Yes.
Okay. All right, now this will be pretty quick. Just a summary of the phytoplankton data for 2020. It was not an exceptional year. It was a good year. The Denaliella, the chlorophytes were high throughout the year, which is generally good for for shrimp production. So that may be in part also why the cyst production was better than we anticipated, because the food was better. Also, we can see here that in 2020, the blue line down here of the chlorophyll, it got depleted, as we talked about in August and December. It had been depleted by the shrimp to a much greater extent than we have seen on average over the GSLEP study, but it has recovered as of December. Now, I'm not presenting here, I didn't present here the January or February data from this year, but if we look at that February value for 2021, it's high compared to what we have been seeing in recent years. And so the phytoplankton certainly have recovered from that January dip that was reported. So this value right here is above what we've been seeing, say, over the last 5 years on average in the lake. So that bodes well that that January decline is not going to have much of an effect. As Kyle pointed out, it was a strange year in terms of sort of the timing and the peaks that we saw in the different categories of brine shrimp. However, again, the adults were not that far off, but we saw a huge increase in the juveniles, unlike the average, the red line here, and a good. We didn't get the GSLEP counts on the nauplii seemed to be missed in the spring, the peak that should have been there. So we had it looks like there was a early high hatch, got a lot of juveniles, that depleted the phytoplankton in the lake, and that led to a somewhat, but not an atypical decline or lower number in the adults in the lake. So there's really not much more to go over the 2020 data for the lake. What I really want to focus on today is what we've been doing with some of the experiments in the lab and how this relates to the modeling of the Great Salt Lake. First of all, we have in the lab got all of the GSLEP data is available for GIS analysis. The data are all entered from all the way through 1994 to present. However, the data still needs to be collated between sites and dates, which is being done. Then those data files need to be quality checked to be sure that all the data are correct. And this has all been completed for the last 5 years, for 2015 through 2019. And we're in the process of working backwards from 2015. And basically that's being done. Each data set for a year is being given to an undergraduate who is entering or collating the data, and then Andrea, who I think is online for the talk today, my lab technician, She is then quality control, examining what the undergraduates provide for each year. But to give you an example of what this GIS data allows you to do, here's a sum of all the cis, all the nauplii, and all the females counted at all of the stations from 2015 to 2019. And if you look at this, it is quite obvious particular locations in the lake have the highest productivity in terms of cis, highest productivity in terms of nauplii appearing, and then the highest numbers of the females that are present. So there definitely appear, for at least the last 5 years, very consistent levels of production at different locations within the lake. Now, the other thing that I talked about in August and in December is that we have completed now all of the phytoplankton responses in laboratory studies, looking at the maximum peak amount that is produced, the time it takes to go from a very low initial sample, inocula, of the type of phytoplankton, and the rate at which they're approaching that maximum value as a function of temperature, salinity, nitrogen, and for the bacillariophytes, the diatoms, the effects of silica. So we have all this data now, which we didn't have before. In the past, we were working primarily just for an average sample from the lake. We also now, from the laboratory, have the harvesting rates, the grazing rates of each of the age categories, stage categories of brine shrimp on the different types of phytoplankton, chlorophytes, bacillariophytes, and the cyanophytes at different salinities, temperatures, and abundances of each of those types of potential foods. Something that is totally new that I haven't presented before, and this is because this was an experiment we started last year, and we had problems running this experiment in part due to COVID. The first experiment we looked at was what happens if, well, what we were looking at was what happens if you mix one type of phytoplankton with another type of phytoplankton. Are the brine shrimp capable to selectively graze the different types of food? So we mixed them in equal abundances, the two types of phytoplankton. We allowed the brine shrimp to feed, and that would be 1 adult, 2 juveniles, or 10 nauplii, each by themselves. Allow them to feed for 1 hour, and we knew the cell counts before and the cell counts afterwards. The difficulty, the hard part of this experiment is that you can't use the fluorometer counts, because the fluorometer count just measures the mixture of the two foods. So you actually have to do the cell counts and identify it to what food each cell belongs to. Now, the problem we ran into in starting this experiment last year was we started with Dinelliella, the chlorophyte, mixed with Nitzia, the diatom, bacillariophyte. And the first experiment that we ran was the day before the university shut down for COVID. So we put all of the results after the 1 hour, we fixed them, and we weren't able to start counting them probably for 2 months or more afterwards. And we got some really strange results in that the Dinelliella went way down in the preserved samples, and we then had to go back and do another experiment to see whether the preservative was working well with Dinelliella. And over that extended period of 2 to 3 months, we found that Dinelliella is not preserved. Diatoms are, thanks to their silica, but the Dinelliella is not. So we then reran that experiment and then ran into additional problems when we found that the diatoms probably are sticking to the sides of the tubes and therefore biasing the cell counts that we're doing. So at that stage, at this fall, we redid the experiment, this time with Denaliella combined with the cyanophyte, Coccicloris, or Euhalothaceus. There's another more common name that's used for it now. It's changed, but Coccicloris. And the results came out very interesting. First of all, after 1 hour of feeding, we found that from the controls, no shrimp being present, we had an expected relative abundance, and this is presented for Denaliella. We had an expected relative abundance. Then we had the observed relative abundance when there were shrimp present, and you can see that the relative abundance of the Denaliella went down much more than expected based on the controls, indicating that the shrimp were preferentially feeding on the Denaliella over the cyanophyte. Then, to show how much they preferred to feed on the Denaliella, we knew from our feeding experiments, where we knew the rate at which cells were ingested when you gave the shrimp only the Denaliella to feed on, or only the cyanophyte to feed on, we had what we would expect to see as the percent reduction in the Denaliella cells. That's because it's very low, because the cyanophytes are harvested in numbers of cells per unit of time at a much higher rate than the Denaliella are harvested in terms of cells per unit of time. When we looked at what the ingestion rate was of the shrimp, we found that their ingestion of the Denaliella was much higher per cell per unit of time. This is clear evidence that the brine shrimp are not just randomly filtering the water that they encounter. They are actually selecting out of that water particular food types, phytoplankton types. And this means that their feeding, when we go to model it in the lake, cannot be treated as just they're going around and randomly taking whatever they filter in. They are actively searching for and taking particular foods. Interestingly enough, an early paper, the only other paper on selective, that tried to look at selective feeding by brine shrimp, wasn't able to identify any selective foraging. However, that was done using phytoplankton that the shrimp normally don't feed on. This was a study done back in the 1960s. This, though, is a clear example, and it's highly, highly statistically significant. And you can see here that the shrimp are showing a very high preference for eating the Denaliella at the expense of the cyanophyte. So that brings us to the model and what all of this additional laboratory data that we have now means for the modeling that we're doing for the Great Salt Lake. This is the model that I've been presenting for the last year and a half or so. This is the module for the phytoplankton, and this is the actual growth of the phytoplankton. And all those lines show the interactions of different variables in the model: temperature, salinity, light, nitrogen availability, etc. This bottom piece here is the shrimp consumption. This is given what the phytoplankton are producing, this is what the shrimp are consuming out of that. So that's the original model. Here is the new model for the phytoplankton module. And if you look at this, with all that additional data that we've collected on phytoplankton growth, the actual module, portion of the module dealing with the phytoplankton growth has become far simpler. Instead of having to make all kinds of calculations of how much is bacillariophytes, how much is chlorophytes, how much is cyanophytes, we now have absolute measurements of the responses of each of those types of phytoplankton to environmental conditions. So this model up here, this portion of the module, has become far simpler. However, the brine shrimp consumption portion of the model now becomes far more complicated, because we now know how specific stages of the shrimp are harvesting the different types of phytoplankton. And we know now that that is not just an independent, it now is not independent for each of the phytoplankton, but depends upon how much cyanophytes are out here, how much cyanophytes are here, the blue, and the brown is the bacillariophytes or diatoms. All of this depends upon how much of the preferred food is available. So there are now far more linkages than we originally anticipated in the model. Now, what does this mean to the how good the predictions are coming from the model? The old model really stunk. Did really well in terms of getting the chlorophyll A prediction. Did terrible, though, at predicting how much of the chlorophyll was composed of each of the three types of phytoplankton. Really bad when we looked at diatoms, really bad when we looked at the blue-greens or the cyanophytes. The new model doesn't do quite as well at predicting the chlorophyll values. And in part, the only place where it really doesn't do well is that the phytoplankton do not recover in the fall as quickly in the model as they do in real life on the lake. I'm not sure why that's the case. It could be possible that the brine shrimp module portion of the model, which has not been changed at all yet, it's possible that we have too many brine shrimp surviving late into the fall, and they're depressing these chlorophyll values. That's the next step, and what I'm working on right now is trying to get at improving that portion of the model, especially now that we have more data on the phytoplankton coming that they're consuming. Here's the observed abundance for the last 10 years of chlorophytes. The brown is the cyanophytes, and the blue is the bacillariophytes. If we look at that and then look at the predicted here, we can see that it's matching up. We're doing much better now in predicting how abundant the three different types of phytoplankton are in the lake. We're doing much better than we used to do with the old model. And one of the things I can point out, I just did some calculations today, that the model is looking at the bacillariophytes here and overestimating them. And that's because the data that we included in the model included laboratory data that were given with higher silica levels than the lake has present. And so when I remove those laboratory experiments with the very, very highest silica levels, this blue value here drops down about 25%. And so it becomes much closer to these values that are observed, and the green, the chlorophyte values, go up in response, getting much closer to this here. So the model seems to be doing very well in terms of the phytoplankton module that we've now modified from the older model. And just as an example of how this is translating to the brine shrimp, and we're working on now the brine shrimp portion module in the model. Here is the red brine cyst numbers in the lake. The blue is the observed cyst or predicted cyst numbers in the lake. And you can see they're a little lower than predicted, but not that far, than observed, but not that far off. Where we see a big difference is here in the spring, and there's a reason for that. The model is set up at this point as the females either produce live young or produce cis, either or. And so there's no possibility going on in the model of their mixing production of the two, and that's going to pick up part of this here when that's built into the model. And the other thing that's going on here is that the model is set up that all cis hatch exactly at one point in time in the lake, whereas hatching may occur over a much longer period of time in the lake. And of course, the cis counts can also include a shell that is left behind. So they're going to tend to be a little higher than expected from the model. That's all I have to say at this point. I'd be happy to answer any questions people might have. But that's where we are with everything at this point in time. Any questions?
It sounds like what you're saying or what you found is Denaliella tastes great, less filling.
Yes. Well, actually, it tastes great and is more filling if you look at what the shrimp can do with it. That's the preferred food. That's the one they survive, grow, and reproduce the best on. And so if you think about this now, in the past, what we did was we had just an average value going into the shrimp based upon the chlorophyll that was in the lake. Now the new shrimp module will take into account how much chlorophyll is exactly coming from the chlorophytes, how much is coming from the bacillariophytes, and how much is coming from the cyanophytes. It's broken down, and that should give a far better resolution of what's going on.
Hi, Gary. Chris Bittner. Could
Chris.
How are you?
Good.
Hey, the algal preferences, could that be tied to size?
It could be, but we don't think that it probably is. Because one of the things that we do when we feed the shrimp in the laboratory, they are the only one that could be size dependent is the diatom. They tend to be the largest, and they also tend to aggregate into clumps. And when we do those feeding experiments, we first of all shake them up really well from the starting culture, and we run them through a filter so that only single cells can go through, or mostly single cells can go through. So for that 1 hour time period, we don't think that clumping, at least in the diatoms, is a problem. If you go to the cyanophyte, they're really small, and when they're by themselves, they're being taken up at a really, really high rate, cells per unit time. So, cell size is playing in, but not in terms of the preference.
Thanks.
Heidi had a question, Gary,
see that. Heidi asked, why do you think there are site-specific differences in production of adults, juveniles, and nauplii? That is the purpose, eventually, of getting at the GIS analysis. There's a couple of things that we could hypothesize that could be going on. One of the things that we can look at is wind directions and wind speeds are being built in. Are all of these individuals being blown over, or are the currents taking them over to a particular place? That's one possibility. Another possibility that I think is probably the case, are those sites more productive in terms of the higher quality phytoplankton? And therefore, what you're seeing there is greater production because you've got better food resources there. So all of the phytoplankton data that we've collected over time and analyzed, that goes into these models, goes into the GIS database as well to look for that sort of correlation that might be there. So there's a number of things that we can look at when we have this GIS database. Does that handle it, Heidi?
Yeah, thanks. That's great. I mean, I figured there were physical parameters that were potentially driving it, but that's really, really cool questions to be looking at.
And it's possible some years that the physical characteristics may be more important than the phytoplankton characteristic.
Is there a possibility, I know currents and wind is probably more likely, that, but is it possible that some of those areas have higher concentrations of grebes that might impact adults and juveniles?
Yes, that's possible. And early on, we put, we have the ability, we have the grebe data that's going into this GIS as well. So we have the counts of the grebes at different locations that can go in there. And early on, with 1 year's data set that we looked at, this was several years ago, we did see a response that we didn't see necessarily. The resolution wasn't there to be able to see that the grebes had reduced the shrimp, but it looked like over different time periods that we had that the grebes had moved elsewhere after feeding at a location. So I can't answer that directly, but that's where we're at right now.
Yeah, I was trying to remember. It looked like a couple of the spots where there were low numbers were around Fremont and on the along Antelope Island, which would be where they were. But
yeah, but remember what I showed you there was the total numbers over 5 years.
Right.
So it is not month by month when the
right
would be there. I mean, we can break it down month by month and then look at what does October in each year look like, or something like that. We just had that as a quick way of showing that there are some patterns there already.
Hey, Gary, this is Thomas.
Hi, Thomas.
Hey, I was wondering, it might be interesting to look at that spatial distribution before the new breach was put in and after, because I think it may have significantly changed the dynamics of the currents in the lake.
Yeah, we can certainly do that once all of these data sets are collated together and quality checked. You don't want to go through these GIS mapping and spatial statistical analyses until you know your data is clean.
looks like Bonnie and Krishna both had questions in the chat box also.
Bonnie, do you know if the diatoms are Navicula, same genus we find associated with microbialites? That's an interesting question, Bonnie, because these diatoms that we raised are Nitsia. And when Kate did her diatom work, she got Nitsia instead of Navicula. And so that's an interesting question. Why are some of the microbialites Navicula and some of the microbialites Nitsia?
Yeah, and the ones in Shark Bay report Navicula as well.
Yeah, I know. I'm just Kate's analysis, genetic and molecular analysis of the phytoplankton. They're not phytoplankton, but of the primary producers. She got Nitsia as her molecular components.
Okay.
Navicula was there, but not.
Yeah, I kind of remember there was a different one. That's what I couldn't remember.
Yeah, that's in her PhD dissertation.
Yeah, I remember that. Thank you.
And we cultured Nitsia as well from the open water, which is where we get our stock culture.
Mm-hmm.
Krishna, do we have any statistical tests for the model performance, observations versus predictions? Yes, we do. I've just done some correlations, and the correlations as of present they were not significant in the old model for the species or the phytoplankton composition. They were statistically significant for the chlorophyll predictions over the months. They're statistically significant now in both the new model versus the observed for all of those things. So we're better off in that respect than the old model. And now what I'm talking about where it's not looking as good, I'm nitpicking away at essentially what's going on there. I can point out where we need to still improve the model, hopefully.
Thank you, Gary, for the clarification. So, my curiosity is, suppose we couldn't find any statistically, significant relation, then, what's your plan to address that problem? Are you going to change your all modeling approach? That's, interconnections or something like that? Or just for curiosity, it's very complex modeling, I know.
Yeah, I mean, where the model right now is, like I said, it's statistically significant for all of the things that I showed. I think that there still are some probably fine points that would improve the fit. And these are fine points that we have the data from laboratory experiments, but we haven't included it yet in the model because I didn't want it to make the model more complicated than I had to. One of the things that we had to include now in the model is not only how productive each of the phytoplankton are by themselves. Once you put them in the model, you had to include, but they're not just going to produce what they would by themselves. They potentially are competing with each other for resources. So the model is built right now that the three categories of phytoplankton are competing for nitrogen, and their ability to compete is built in the model based upon their ability to take up nitrogen when they're by themselves. So the chlorophytes can take up the nitrogen very rapidly. We're assuming that makes them competitively superior. The diatoms take up the nitrogen much slower. And if you look at that ratio, that's what we're assuming, that nitrogen uptake is related to their competition. The cyanophytes pick it up even slower, and that's why they're at a bigger competitive disadvantage. So that's kind of the standard plant population ecology competition type of approach. It's the uptake rate of the limiting nutrient, and we're assuming nitrogen is the limiting nutrient. Does that answer?
Yes, sure. Thank you.
Okay. Anything else?
Other questions? All right, well, then I guess that concludes our presentations.